Kia ora koutou, and welcome to Virtual Brain Week. My name is Xiao-Wen Yu, and I'm a research fellow in the lab of Associate Professor Joanna Williams at the Anatomy Department at University of Otago. Today I'm going to talk to you about my research on astrocytes and a little protein called SAPA. Together we hope that we might bring a new approach to potentially treating Alzheimer's disease. You've probably seen some images like this before, where on the left we have a healthy brain. You see that there are lovely folds in the brain, and there's not much black space, either in the middle here, which are our ventricles, or around the outside between the skull and the brain. However, in comparison, our Alzheimer's brain, we have a lot of dark space, so more space in the ventricles, and our brain matter is not taking up as much space within the skull. That is because the neurons have died off. That's why we call it a neurodegenerative disease. However, this is a situation where we're quite late in the process of the disease. The neurons don't die off at the very beginning. So when we think about the disease, it's no wonder that we're paying attention to our neurons, because even though they're not dying off in the beginning of the disease, they're not functioning as they should. Here are two cartoons depicting very simplified neurons. A neuron's job is to take information from one end and send it through to its neighbor. And it needs to do this accurately and at the right time. And they achieve this through structures called synapses. So if we zoom in, you see that the two neurons make almost contact, and there's this little gap in the middle. And they talk to each other across this synapse. And it is actually these synapses that are lost early on in the disease. So you can imagine that if the contacts aren't working correctly, that the information that's supposed to come from one place to another will not be transmitted, and therefore our brains aren't working as they should. However, neurons are actually not the only cell types that we have in our brains. Think back for me for a second to the last movie you watched. I bet you paid the most attention to the actors that were on screen, because they were the ones who you could see and hear. But if you think about the movie as a whole, they're not the only ones who contributed to make the movie what it is. They had a whole massive support team of directors, writers, producers, even the catering people or makeup artists to allow them to do their jobs. And the brain is no different. In the brain, the neurons, who are our actors, have a massive support crew. And one type of cell that forms the support crew is the astrocyte, depicted here as the green cartoon. One estimate suggests that in human brains, we have 5 times as many astrocytes as there are neurons, so they must be super important. In fact, they're positioned just right to help support neurons talk to each other. If we zoom in, the astrocytes actually send processes to wrap all around our synapses, and they provide lots of great support to our neurons. For example, they give the neurons nutrients like glucose and oxygen to provide the energy they need to talk to each other, and they also take away the waste that's not needed or to turn off signals as they should be. What we've learned recently is that these astrocytes are also impaired in Alzheimer's disease. So we can imagine a situation where the neurons are not talking to each other as they should, but also their support crew is not functioning as they should be. So we have a double whammy situation. Or perhaps it's the support crew that are impaired first, and that's what causes the loss of synapses. So what can we do about this? The Williams lab studies a little protein that we've cartooned here in blue called SAPA. The long name is secreted amyloid precursor protein alpha, but we call it SAPA for short because the other one's a mouthful. It turns out that SAPA does lots of great things for brain cells. First of all, it protects our neurons from toxic insult. It also helps our neurons to talk to each other across those synapses. And importantly, it can stimulate our brain cells to make new proteins, and this is crucial for making new memories. Studies have shown that levels of SAPA are actually decreased in Alzheimer's disease. So minimally, we know that SAPA is something that is very important for normal, healthy functioning, but it's also very beneficial when we introduce it back into a system, whether it's healthy or diseased. Now, I mentioned that SAPA stimulates the synthesis of new proteins, and we can use this as a readout of SAPA's function. So, within a cell, we have a protein machine called ribosomes that runs along this chain of mRNA, which is its instruction sheet. And the mRNA tells the ribosome which amino acids to build into a chain that turns into our growing protein. So what we can do is add an amino acid to cells that has a little tag on it. The amino acid behaves just as your regular amino acid does and gets incorporated into the newly growing protein chain, but because it has a tag on it, we have a handle with which we can see the proteins. So the data ends up looking something like this. We can stain our wells of cells, and in purple here, we have our neurons. We can then stain our astrocytes in red and our newly synthesized proteins in green using the tag that I just explained. And when we look at them all together, the merge looks something like this. And because microscopes and technology have improved so much, we're now able to zoom in and see these cells without losing any resolution, even though we've imaged a whole dish of thousands and thousands of cells at once. So we can then measure the amount of protein synthesis that has occurred by looking at how bright this protein synthesis signal is, and we can even do it for our different cell types, in this case, neurons or astrocytes. So one important question that we always want to ask is, how does this work within the cell? So we might hypothesize that our SAPA molecule binds to a receptor at the cell surface, and that turns on a component within the cell. We'll call it component A. This then turns on a second component B, which finally gives us the output of protein synthesis that we observe. So the first thing we would do is treat two different dishes of cells. Well, in fact, we wouldn't treat one of them, and that would give us our baseline control levels. We would see, without treatment, how much protein synthesis do we see in a normal dish of cells. We then compare that to a dish of cells that has been treated with SAPA to see how much more protein synthesis do we get. Now comes the fun part. We can take advantage of inhibitors or blockers against the components that we think SAPA is working through within the cell. So if we prevent component A from doing its job, even though SAPA is on board, what happens to the levels of protein synthesis that we measure at the end? If component A is in fact part of the process, we might see something like this, where the protein synthesis levels drop down to something that looks like the untreated control group. Because even though we have SAPA on board, it's not able to signal all the way down to cause the protein synthesis. On the other hand, we might be wrong about our hypothesis. They're not always correct. So if we block component B and we find that protein synthesis levels are just as high as SAPA on its own, That suggests that perhaps component B is not important for this process. So we might revisit our hypothesis. Perhaps component A works straight to turn on protein synthesis, or maybe it works through a different intermediary. Either way, we're seeing that component B is not involved. So we have a pretty good handle of what these pathways look like in our neurons. But current work and ongoing work is trying to figure out how this signaling is working in astrocytes. And to date, most of our work has been in rodent brain cells because logistically it's much easier to get hold of more of those to work on. However, we are taking some exciting new steps for the next part of the project, where we will be looking at human astrocytes. So in this case, we'll be using fibroblasts that have been donated by participants, and we have the technology in place now to be able to convert them into astrocytes. And we will have astrocytes of two different kinds. We'll have healthy, normal functioning astrocytes, and astrocytes that have mutations which lead to early onset Alzheimer's disease. So we will compare these two. That allows us to ask two very important questions. One is, how does Alzheimer's disease impair the function of astrocytes? This gives us very important information about astrocyte function and how it might progress in the disease. And two, can SAPA treatment then fix these issues? If so, that would be the first step towards potentially a new treatment for Alzheimer's disease, which I think we can all agree is not only much needed, but very exciting. So with that, I would like to thank my mentor, Associate Professor Joanna Williams, the Department of Anatomy and the Brain Health Research Center here at the University of Otago. A big thank you to the Neurological Foundation for funding this project, without whom it would not be possible. And last but not least, a big thank you for your time and attention. Kia ora.